Our next speaker is actually found by Trace Ann Rose, who was a big part of the NF Forum for many, many years and has moved on from CTF. But I'm very excited to invite up Kevin Freiert. I hope I'm saying your last name correctly. Okay. Kevin has over 35 years of experience in biopharma industry and has spent most of his career at Pfizer and been part of the development of 10 approved medicines. And in 2017 started his company Salem Oaks, which is dedicated to helping patients and caregivers shape the future of medicine. And I'm going to stop there and let him take it over for us. Come on up, Kevin.
Thank you, Kate. I'm going to get a drink of water before I get started here. Nice to take the mask off and have that orgasm, but so thank you very much. Yeah, my name is Kevin Freiert. I spent 30 years at Pfizer in many different roles. I started in discovery in the lab, so doing bench scientific work. I worked in regulatory affairs as a medical writer. I eventually became the chief of staff for the head of worldwide development. And while I was working for him, invented something called Pfizer Research University, where we taught people who worked in this big complicated system how it works. You know, that's my gift that I have, is I put together the big picture so that people can see what they need to do in their jobs to meet their most important goals. I started Salem Oaks when I left Pfizer because I realized patients were being engaged with without being prepared to do that. And if you know what you're engaging with and a researcher comes and says, hey, I'd like your help, if they have to spend all their time with you trying to teach you what it is they're helping with, they're not actually going to get their work done. If I could step in and help you understand the bigger picture, I might be able to help you have a bigger impact. So this is my disclaimer. I'm speaking for myself and my company today. I'm not speaking for any of my former employers, and I'm not speaking with any of the companies that I consult with today. And I'm not going to be giving any medical advice here. In fact, it would be silly of me. I know so little about NF and schwannomatosis. I can say those words, but it's only because I've been studying for a couple of weeks. You guys are the experts, and that's why I'm going to stay for this whole conference, because I want to hear, I want to learn from you, and I want to understand deeply what's going on. So the objectives for today, what you'll be able to do when you leave, is describe the purpose of the various stages of drug discovery and development. You'll be able to identify and see the diseases or the decisions that are made there and evaluate those decisions and identify the areas where you can have an influence. It's great to say, hey, we'd like your help. Or you may say, I want to get in there and I want to tell them what to do. But if you're asking the wrong questions, it's just going to bounce off and it's not going to be as effective as you'd like it to be. So we're going to talk about some of those. What's great is the first two talks nailed a bunch of them already. So the flow of the session, I'm going to go through the drug discovery and development process 2.5 times, I would say. The first time is going to be kind of the what and when. What's going on? What happens? What are in the steps? Probably a little more deeply than you've seen before. Let me ask first, how many of you are patients and caregivers? How many of you are researchers? Oh, you guys are going to call me to questions later, I'm sure. Then I'm going to go through it again, but we're going to look through a different lens. We're going to look at what are the decisions being made along the way. You can't influence the science. The science will give you data that will give you answers. You can influence decisions because those are made by people. And then I'm going to talk about those points of influence. So let's define this first. Drug metabolism is a multifaceted scientific process that helps us learn about a chemical or a biological molecule so that we can figure out if it is legitimately, and we can be confident in finding out if it's legitimately and legally a medicine. So let me ask, how many of you would take this if I told you it's a great medicine? You know, there's a lot to think about here. Well, what's it a great medicine for? How much of it should I take? How often? Oh, and where am I going to store it? And when should I get rid of it? And will it affect my other medications? Back in the 1800s, people used to drive the wagons around and sell snake oil. And they'd say it would do marvelous, wondrous things. You can even see on this label, it says it relieves instantaneously. And then it has a whole list of things. Well, in 1906, the government stepped in and said, you know what? We've got to do something about this. And they came up with the first drug act here. So it's the Pure Food and Drugs Act. And one of the key elements of this is you cannot misbrand a drug or a food or something that people are going to take into their bodies. Misbranding relates directly to the label. The label becomes a critical piece in all of our regulation of all of our. Were you just asking me to speak up? Oh. So, excuse me, it underlies all of the regulation now. The label is central. It's critical to what happens in R&D. In fact, I would say that all of the research and development that goes on produces one thing. It produces a label. That's the thing that comes out the end. Sure, there may be a drug involved. There may be a treatment, a gene therapy. It's the label that's the product. That's what we're building. And what is it? It's the accumulated knowledge of all the experiments that we've done with that particular potential medicine. It's all documented. It gets reviewed by the different authorities in the different countries. And then it gets condensed down into what's called a package insert or a label. I'm sure you've all seen that when you get a prescription and you pull out that piece of paper and you cannot read the type, it's so small. That's actually the instruction set for doctors and other prescribers. Here's how to use this. Here's how often to give it. Here's who to give it to. Here's who not to give it to. Here's some things you could expect when somebody's using it and how to manage those things. So the label becomes critical. So how do we get there? How do we actually build this? This is the classic drug development process, and it's just my representation of it. I'm going to go through this kind of quickly, and then we're going to cycle back and go through it deeper and deeper so that you can see more. So in discovery, this is when you first have an idea and you have a hypothesis about how you could change a biochemical activity to change the course of a disease. And so you start looking for a molecule to do that. You take that molecule, when you find something, you go into preclinical safety. And for preclinical safety, what you're trying to do is make sure this is something, we could give to humans. We need to know enough about its safety before we can give it to human beings. Oh, and by the way, when you first find a molecule, you probably have 20 or 30 micrograms of it. In order to do human experiments, you need to be able to make kilograms of it. That's 1 billion times more. So somehow you need to figure out, how did they make that little scraping of a new chemical and turn it into something that could be actually processed into a drug? Before you can go into phase 1, you have to talk to the regulators, get their okay to go forward. So we have something called an investigational new drug application, or IND, in the US. Other countries call it a clinical trial application. It serves the same purpose. It gives you the authority to run these clinical trials. Phase 1, and I'm going to talk about this as for typical diseases and then reflect on rare diseases and a little bit on NF as I drill down. But what you do in phase 1 is you typically go to healthy volunteers and test it to see if it's safe, but also test it to see, what does the body do to that drug? How does it take it in? How does it move it around? How does it get rid of it? If you get through phase 1, you go to phase 2. And in phase 2, you finally get to test the hypothesis that you had way back in discovery, because you're going to start testing it in patients who have the disease in question. If you prove that concept and the hypothesis looks like it's holding with that molecule, well, now you're going to look for the best dose to take into phase 3. And you're going to narrow that down to 2, at most 3 doses that you would take into phase 3, because phase 3 is hugely expensive, and every dose that you test adds, another arm to the trial. And then you have to have your controls in there too. It could be a placebo control, or it could be an active control, something that's already approved for the treatment of the disease. Once you've done phase 2 and you've selected your doses, you move into phase 3. And the purpose of phase 3 is literally to confirm phase 2. You're trying to show that we had an hypothesis, we tested it in a very narrow range of people. We want to see if it works in more people. We want to see if we can make it work again, that it wasn't just a one-off thing. But you're really casting a net for safety. You want to expose enough people to the new molecule so that you have a really good idea of what to look for if and when it gets out in the market. Phase 3 runs, phase 3 will be longer, because you want to see the long-term effects of what the medication is. The proof of concept may have been a couple weeks long. Phase 3 can be years long. Once you've gotten through phase 3, you go to registration, you file to these regulatory authorities. Another acronym here, the new drug application, it goes to the US, or a market authorization application goes to the EU and most other countries in the world. Japan has a JNDA, they've copied us. That's the legal authority now to sell your product. Now, biopharma companies are for-profit institutions. They need to be able to sell their products at the end of this. What I haven't shown here is that for the products that, I can't step away, can I? For the products that make it into preclinical, the drugs that make, or the molecules that make it, only 4% actually become medicines. So it's a 96% failure rate. What would your boss think of that? So let's go a little deeper here. I'm going to break it into 3 stages so you can kind of see how we think about things. We have research, development, and registration. Research it includes discovery, preclinical, and phase 1. Oh, darn, I didn't want to click that yet. So these 3 stages in there take 6 to 10 years. 6 to 10 years and several $1 million. It sounds expensive, but it's the cheap end. The thing that's really worrisome is the time, but it's somewhat justified and somewhat understandable. So there's 4 steps in discovery. You can read all this, but basically what you do is you come up with an experiment that you can repeat over and over again. You put millions of compounds through that to see if it shows something, just something. It's not very quantitative. It's just kind of does it make it change color? That level of experiment that you're doing. You find a couple dozen of those that works, and you take those are your leads, and you start to do chemistry around that. And this is the same for protein molecules or monoclonal antibodies, the biologics that are out there. You start doing work around it to optimize it. And what you want to end up with is 1, but probably 2 or 3 compounds that you move into preclinical. Why would you want 2 or 3? Well, because 96% of them fail. So you want to have a backup and maybe a backup to the backup. So you don't have to go all the way back to square one if the first one fails for some reason. So why would it fail? Well, that's what preclinical is about. One thing you're doing is, as I mentioned, you're starting to create clinical supply here. You're starting to build up the drug supply from just a test tube's worth to, barrels of it. But the real intent of preclinical is safety. And the mindset of people in in preclinical safety is, let's get rid of the bad ones. We're going to weed out the dogs and get rid of anything that doesn't meet our criteria for what is safe enough. And we do some early pharmacokinetics. I'm going to explain pharmacokinetics in a minute. Basically, it's what happens to the drug when it goes into a body. This could take you another 2 or 3 years. You go to the regulators, you file your IND or CTA, and you do your first in human study. These are done very carefully, very slowly for what you're doing. The first study, literally the first time a human being takes this brand new molecule no one's ever seen before, they get 1 dose. And you'll take a group of 8 or 10 people, some of them will get the drug, some will get some placebo, and you'll give a very low dose. And then you'll give a higher dose if that's okay in a different group of people. And then you'll give a higher dose in a different group of people, but just 1 dose per person. Because if something goes wrong, you want to see it right away. You'll also look at pharmacokinetics, which I said, I promise you I will explain that. It's a big, long word, but it's pretty easy. And pharmacodynamics, which is easy to explain, which is, does the drug work or not? Does it do something? Does it change the enzyme we thought it would? Does it change the cellular structure like we thought it would? And so you're looking at very early signals, but remember, this is usually done in healthy people who don't have the disease. So it's really not telling you what's going to happen in those diseased folks yet. If you get through this single dose study where you're escalating your doses or ascending your doses, then you go into multiple dose studies where you'd have 2 weeks of dosing. Again, small group, low dose, go up a bit, a small group, low dose, small group, low dose, or a slightly higher dose, until you get to what you call your maximum tolerated dose. When you start to see something that people saying, I wouldn't do that again, or I won't keep taking it the whole 2 weeks. Okay, let's go back down to where people would take it. So you're kind of feeling out the boundaries here of what the window is that you can use this drug in. So pharmacokinetics. I want you all to imagine that you just took a pill. Where does that pill go? It goes to your stomach and it might get absorbed there, or maybe it waits till it's in the intestine and it gets absorbed there. Once it's absorbed and it's in the bloodstream, where does it go in your body? Well, the blood goes everywhere, but the drug may not. It may not go to all tissues. Particularly the brain doesn't always get exposed to the drug. There's a blood-brain barrier there. It's hard to get drugs into the brain. So you need to know, where is it being distributed to? Well, once it gets distributed, the body's going to say, we got to get rid of this stuff. This isn't us, this is something else. So what happens to it? It gets filtered out by the liver, and the liver is full of enzymes that has all these different tricks for metabolizing these drugs. It can add something to it to make it more soluble so that it leaves the kidneys. It may chop it up a little bit into smaller pieces that's easier to excrete. So I just mentioned the 4 parts of pharmacokinetics there: absorption, distribution, metabolism, and excretion. And pharmacokinetics is the science of studying how fast do those things happen and how do they happen. Why is this important? Well, how do you think we figure out what doses to give people, and whether it's a 1 a day, a 3 a day, once a week? I mean, I've seen drugs now that you take once every 6 months. That's all pharmacokinetics that is informing that. And that's the key purpose of the phase 1 studies, which takes us now into development. And this is phase 2 and phase 3 are the major development portions. Phase 2, I talked about proof of concept. You finally get to test the hypothesis you came up with in discovery 6 to 10 years ago. And if it works, and you show that it works, you usually put it in kind of a higher dose there. You select a dose that's pretty likely to show something. You just want to test the hypothesis. If it works, then you go into looking at your doses and what's the optimal dose to get people. How do we balance the safety findings that we see, the issues with it, with the benefits that we see, and what's that sweet spot? And you probably start thinking about, well, what do we want this drug to look like? Think about all the different ways you can take a drug. I used, you swallow a capsule or a tablet. Those are 2 formulations. You can have it injected. It can go into your muscles, your veins, under your skin. Oh, there's another 3 formulations you'd be thinking about. What about like a topical, something you rub on? Anybody ever use eye drops? How about an inhaler or one of the things that goes up your nose? There's many different ways that you can get a drug into the body, into the right spots that you need it. I love that high frequency ultrasound thing where you put like something there and then you blast it and it opens up the drug right at, in the site that you want it. That's fascinating. That's like futuristic stuff. That's wonderful. The other thing you do in phase 2 is you learn how to run a trial in this population of people. What should we be measuring? How long does it take for it to move? What kind of movement should we see? What's the variability? All of these things all lead up to designing a phase 3, which confirms what you saw in phase 2, but also gives you all your labeling claims. So you're going to confirm what you saw in phase 2, and you're going to really cast a wide net for safety. You're going to look at adverse events. You're going to look at serious adverse events, which are deaths, hospitalizations, birth defects, cancers. You're going to look at lab tests, blood tests, or urine tests, and you're going to look at long-term effects. There's something to think about in phase 3, though, for you guys to think about. Whoever you study in phase 3, those are going to be the people who are approved to use this drug when it's out on the market. That's who it can be prescribed for according to the label. Doctors have some flexibility. They can go outside the label. Guess what? Insurance companies don't like that. Access becomes a problem if it's not in your label. So you need to think about phase 3. Is it going to capture everybody who's could benefit from this drug? And can it still be a good experiment that actually shows this drug is doing what it was intended to do? Registration, I'm going to skip through really quickly because I've hit it already, but you need to get regulatory approval to go into clinical trials, and you need to have regulatory approval to get your market authorization. You also have to update the regulators on an annual basis at least. If something goes wrong, you need to update them right away. A serious adverse event needs to be reported to the FDA in 24 hours from when you heard about it. It's that fast because it's a death, a hospitalization, a birth defect. There's 3 major regions. My only point here to make is that the US, the EU, and Japan have been working for 25 years to make this simpler. It's the same general process they go through, the same requirements, but actually they've come to create something called the Common Technical Document so that companies don't have to create 3 different applications. They can create one and use it over and over. Japan will ask for a few more pieces. The Europeans don't want to see all the raw data. That's okay. It's one document. You get what you want. That's made it much easier. And it's made it much faster for companies to be able to do the work and the regulators to be kind of in sync when they do things. And then you've got the rest of the world. And here's probably for most people that you don't see, every one of the countries in the world has their own regulatory process. Some of them rely on the US or the EU, but every country in the world has their own process. And when a sponsor takes a medicine out there to get approval in all these countries, they've got to deal with that. That's part of the work that you do. So summarizing the process, it goes back to my definition. It's a multifaceted scientific process. Learning about a molecule. Remember, the powder was nothing. It's all the stuff we learned about the powder that makes it a medicine. And that allows us to really call these medicines. You'll hear me stumbling over drug, molecule, compound, medicine. That keeps me awake if I keep changing it. If it confuses you, let me know. But really, it's not a medicine until it's been approved. So we're going to shift now to decisions. But first, I just want to put up there that basic process again. I'm not going through it again, we did that, but we're going to look at it a different way. I want you all to put on your decision detectors here, and we're going to watch a video, and I hope this works. I didn't check with the AV guys. Will this work? Oh, it's doing something.
Before it became a medicine, it was an idea, an inspiration, a wild what if. So scientists went to work. They examined 87 different protein structures, had 12 years of setbacks and breakthroughs, 4,423 sleepless nights, and countless trips back to the drawing board. At first they were told no. Well, maybe. And finally, yes. Then it was 36 clinical trials, 8,500 patient volunteers, and the hope of millions. And so after it became a medicine, someone who couldn't be cured could be. Me.
So what decisions did you guys see there? I told you to put those decision detectors on. Anybody, just call them out. What'd you see?
Even just the will to try.
Wow, that just gave me chills. Yeah, the will to try. Imagine if 96% of everything fails, how few people actually get to work on successful products. That's why I put in my bio there that I've been on 10 of those. When I was in the labs, there were people who spend 40 years and never are on a successful product. And they love their jobs and they come in every day and they fight harder and harder and harder. They're at the beginning of the process, so they have to do that. They're driven by something different than getting the product on the market. A lot of them are driven by science itself. A lot of them are driven by the challenge. Every one of them that I've ever met is driven by the patient. It's great to come to work and say, I hope I do something today that helps people. So we kind of looked at, well, what decisions are there? And there's a ton of them. And I didn't even capture everything that's there. And I've got a lot of background going in my head when I watched that commercial. And I didn't want to do a Pfizer commercial, but it's just such a great video. It really captures it, especially that pizza part. I remember those nights. But there's different kinds of decisions that are made, different levels of decisions. There's a lot of tactical decisions. What color is it going to be? How big is it going to be? Where are we going to run our trial? Our first trial? Are we going to be in Europe or the US? Why do we do that? Tactical decisions. The bigger decisions, strategic decisions, the go and no-go decisions are where that 96% failure comes from, because somebody has to decide that one's not going. It didn't make the cut. So we're going to talk about some of those decisions. We're also going to talk about some of the decisions that are more relatable for what you're thinking of. How do we keep this research moving? Some of the things where I think you could have huge impact. So this is the same drawing, except instead of steps in the process, now it has all the decisions that are going to be made. I'm going to go through each of these, so I won't bore you with my attempt to try to be concise here. It never works, so I'm just going to skip through the slide. I think you'll have the slides afterward to be able to go and look at it. So the first team that works on this is discovery team. Discovery teams are relatively small. A few labs get together and they're working on something. And so the input to their decisions is a scientific literature, expert opinions, which actually reminds me something that Cynthia said before, I think it was, is around the Registry, where do we find these patients? Heck, where do you find experts in rare diseases? I was working on a rare disease product. There were 7 people in the world who treated patients who you could call an expert. And we were overloading them just with our work, let alone their practice, let alone other companies trying to do research as well. So expert opinions, and then their original work. They do a lot of experiments, tons of experiments. And the decisions they make are, which approaches should we pursue? Should we go with small molecules? Should we go with a biologic? Which enzyme, layer should we hit here? Koselugo uses, it's a MEK inhibitor, which is like the 4th step in a long chain of steps. Somebody decided that's the one to hit, right? Which compounds do you move forward? So I told you, 96% of them don't. So at the early stage, they have to say, what is a hit? What's a lead? How do we do that? And they have this other tactical but business decision, when to file a patent. The clock is ticking as soon as your patent is issued. You've got 20 years to do 12 to 15 years worth of research and development, and then hopefully sell a product that will pay for just itself, but also pay for future products. So if you file the patent late as possible, you'd have as much time at the end, right? But if you file it too late, somebody else may file one first and take it all away from you. So this is a very interesting, it's a gambling feeling of when do we file that patent? And all companies have sort of patterns of how they do this. And as knowledge happens faster and science happens faster, it's happening earlier. So that's a decision this group makes. The next group is the preclinical team. The discovery team kind of fades into the background a bit. They don't go away. They're still there to give input because they can tell you about the molecule and about the experiments they did. And they're very, very interested in what these people are going to do now, because they're going to take data from animal models, and their mindset is get rid of the bad ones. They come in and they say, we're going to kill projects. That's our job. We are the big filter here. I can remember talking to a very senior person in the drug safety evaluation, and he said, our job is to protect the company from itself. And so that's what they do here. That's a different mindset than let's go find drugs that are going to work. But do you want these people left out? No way. These are the people you want standing in the way saying, nope, we don't send bad stuff through. And then they ask a question. They answer a question. They make a judgment. Is it safe enough to go into humans? I want you to focus on that term, safe enough. Safe enough in whose eyes? Those guys in the white coats, they may have one version of what safe enough looks like. I don't know if it matches what your version of safe enough looks like. What is the toleration there? So think of that as an area that, gee, if we could let them know that somehow, what our risk tolerance is, then maybe they'll make decisions that are consistent with what we would do. The other half of the preclinical team is the one that's working on the drug and trying to figure out how do you make it from micrograms into kilograms, and how do you turn it from a powder into product that you can take, capsules, tablets, lotions, whatever it is. So their decisions are, how are we going to do this, and which formulation will work best. Again, I don't know NF enough, but I do know that you get the tumors, and I don't know what the best way for you to take a drug is, what's comfortable for you, what's not. That's another area that patients can get involved in quite early in the process to say, this is what we'd like. So now something scary happens. The decision-making goes out of the sponsor's hand and into the regulators' hands. And the regulators, they're only getting what the sponsors give them. That IND and that clinical trial application is the accumulation of all the data that the company has produced or the organization's produced. And they're the ones signing to say, yep, these people, we can give this to people. Someone's going to take this for the very first time. That weighs heavy on them. So first thing they ask is, did they give me everything? Is this everything I need? And did they ask the right questions? Did they do the right experiments to inform this? Are there any unwarranted risks to administering to humans? And if there are, well, now they make their go, no-go decision that's outside your company's control. And in the US, they don't actually say yes to an IND. They let an IND go into effect, or they put it on clinical hold. They say, we have some questions. We'd like you to do something. There's some more science you need to do to inform our decision. So think about how could you impact those types of decisions as the voice of the patient. So you move into phase 1. They're going to run studies. Typically, it's in healthy volunteers. I don't know if in NF, if they go into patients right away. Somebody just said that there's a lot of oncology products that come through. In oncology, because chemotherapy has tended to be pretty brutal medication, they usually don't put that in healthy volunteers. They go to patients right away, which a lot of patient groups, other diseases are like, yeah, why don't they go to patients right away? Well, you're looking for those safety signals and you don't want to confuse them. So that's why you go into healthy people first. So again, they ask the question, is it safe enough to go to the next stage? Safe enough according to whom? Now, these are people who actually work with patients. They're clinical pharmacologists. They're focused on the science of pharmacokinetics and those things. So maybe they've got a good idea here. And which doses are you going to take into phase 2? What's that proof of concept dose, that really good shot you're going to take? When you move into phase 2, again, they're doing clinical studies. This is how they do their work. And they're going to see their safety signals. And they're going to suddenly get a whole bunch of ideas from the organization around them saying, here's what we want phase 3 to look like. Commercial people may start getting involved now and saying, this is what the product needs to be. That's not the way scientists work. Scientists work like, let's see what the product is, and then you can figure out what it's going to be called and how it's going to be marketed out there. But the inputs are coming in. So they're looking at this, and again, have they proven the hypothesis? Does the compound prove the hypothesis? Did it work? That would be great. Sometimes an even better answer is, wow, we were really confident in this compound. It gets to where it's supposed to. It does what it's supposed to, but it didn't affect the disease. That's not better for you, but it's better for a company because now they know the hypothesis doesn't work. Let's go to a different hypothesis. Let's put our investments somewhere else. So they're not running down dry wells all the time. Again, is it safe enough? Now, these are teams of physicians who treat people with a disease or who have focused on the disease enough to say, okay, here's what these patients need. Does your doctor always know what your risk tolerance is? Does your doctor always know what really bothers you? Does your doctor always speak for you? I'm guessing, with all the experience I have with all the other patient groups I work with, no. They're translating it themselves. And then there's another business question. Is this molecule worth the really big investment? The amount that you spend in phase 3, it multiplies 8 to 10 times what you've spent already. Do you want to do that? And so you want to have a pretty good shot at it. This may be a time to say, oh, let's look at the backup and see which one's actually better, and spend a little bit more time there. What would your viewpoint be on that decision, right? How can you inject it into here and say, wait, I don't want to look at the backup. That's going to take 2 more years. Let's try this now. And if it doesn't work, it doesn't work. And so somehow, how do you influence somebody who's looking at big investment versus, no, we need a drug now? So I want to talk about, I'm breaking my little cycle of the teams and what they do, a little bit about rare disease and oncology products as well. It's called accelerated approval. And this is a mechanism that's been put in place to allow early submission of that NDA. So you go through phase 2, and you're probably looking at a biomarker there or some endpoint that needs to be reasonably likely to predict clinical success, clinical outcomes. And that's a very woolly term when you start talking to regulators about that. And they kind of fight that a lot, because it's like, you're taking the shortcut here, you're taking the shortcut. The first decision is, what does phase 3 look like? What are we going to study? And the second is, what do we need to support the claims that we want to have in our label? So they're thinking about this. Okay, great, we do this design. Once you get phase 3 going, it goes. And the data come back, and you need to make the decisions of, gee, do we have a product here or not? And once again, next slide. Look at that. Now the team got bigger. Once again, it goes out to the regulators again. And the regulators have to decide whether or not this is a product. And the only thing they have to work with is what you've given them. And you have to give them everything. That's the law. If you're studying it in something else, they need to see that safety data. They need to see all the data you have around your compound. And the things they're looking at are, is there sufficient evidence? Did these trials, were they strong enough to believe them? Is the benefit risk ratio acceptable? And do the data support the claims that this company is asking for? They decide whether or not you're approved. And then now you can imagine, I mentioned that most companies are for profit, and they're trying to, maximize their product. They want a label that's favorable. Regulators want a label that's safe for public health. So there's this tension there, and that label gets negotiated. Once again, those are things where people are doing, using their judgment, and you might be able to have some influence there. Next slide. I'm going to quickly touch on what the business managers are doing while all these scientific teams are working. They have to decide which projects get funded. And they don't have infinite money, so it's limited resources, big limits sometimes, but which ones actually get funded? Which ones don't? How much should be invested? What's the appropriate investment at any stage here? And with those ongoing investments, how do you prioritize them when things change? Again, with COVID, we saw companies take their eye off the ball of all the other projects they had and focus on their COVID projects. Move resources, move people, move money to vaccines or therapies. That's great. That's a really big, obvious change in the world that you needed to address. Most of these prioritizations are much more subtle. Does that pain medicine work better than that depression medicine? How do you make those decisions? It's very difficult. Will they be able to know I said that? We are working on the captions. We apologize for the inconvenience. It's just taken a little bit more time than we thought. And then the business managers actually have to decide, what kind of return do we need to see? That sort of sets some goals for people. It needs to be this kind of product. And that helps people make other decisions. You can see the difference in cost there for phase 3. So the summary around decisions, let me see what my time is looking like. I'm doing good. Medicine's development is really just a series of interrelated decisions. They build upon each other, they're intertwined with each other, and they will affect each other. Something that you decide today in your early program might affect a decision you're even able to make 10 years from now. Or let me put it another way. If you wanted to go to a company today and say, hey, I know you have a product that's in phase 3, could you make it do this? You should have asked that question in 2005, because that's when they were actually looking at what they were doing and they were making the decisions to go either here or there. And so that's where they're all tied together. And over space and time, they affect each other. The other important thing is they're made by people, and these are educated people. I used to love sitting around the table with a bunch of people with MDs, PhDs, and then the triple threat MD, PhD, JDs. People that are so highly educated, but you know what? They only have partial information all the time. They never have it all. They will always want to do one more experiment before they make the call. They have biases. They come in with their own preconceptions of what's really needed, or what's important, or what's safe enough, or what works enough. And somebody mentioned, is 20% reduction of the tumor size enough? My guess is that the first answer is, well, no, if you can go farther, go farther. But does 20% actually make a difference in your life? I can't answer that. I don't know you guys well enough yet. And over the next few days, I hope to get to know you more. And all these people have differing objectives and agendas. The regulatory folks are there for public health, public safety, the regulators. There's other people who are there for profit. There's people who are there because it's really challenging science and it's fun to do. There's people there who want to help patients. There's people there who just have a job. So all of that together to me is, this is so ripe for patients to step in and say, this is what we need, and this is what we can do, and here's how we can influence it. What's wonderful is my next few slides, they've already covered most of what I was going to say, so I can move quickly. But let me tell you about an effort that happened several years ago. I think it was in 2013 it started. It's the Clinical Trials Transformation Initiative. It was cross-industry, and it included patients, it included sponsor organizations, it included regulators, included payers. And they said, well, where could patient groups, where should companies try to engage patient groups in this process? I had to say that a specific way. They were trying to give instructions to the companies on how to reach out to patients. Because somebody said it before, how do you find them? When I first stepped out of Pfizer, I was like, wow, I didn't know this was all going on. Why? And I talked to people, scientists who are like, well, we don't know how to talk to patients. And I talk to patients and say, we can't talk to those scientists. They have all those degrees. That's the gap I wanted to jump into. But really, this only goes one way. It's telling companies, here, you can do a program that'll do this and it'll reach out and do it. So I'm going to focus on a few things that CTF is already doing, that you're already doing, and then give you some other insights and encouragement. So we talked about the registry this morning. My big point here is join the registry, just like everybody else said about the registry. Join the registry. That data is very important. But what's really important is the natural history that it gives us. If you can draw a diagram of how the disease moves, if you can see the trajectory of the disease, that can be used in drug development later as a control. It's called a historical control. But if you don't have that data, it's not there. So you're going to do placebo controlled. And everybody's flying blind if they don't know what the natural history is. So somebody said before about and I don't remember what the comment specifically was, but people who are more severe are more likely to be in the registry. You know what? You need the people who aren't that severe, the ones who have just seen their first symptom, the ones who have, the mutations that are likely to cause NF or schwannomatosis, because you need to see what happens in those early days. If you don't know what's happening in the early days, there's no way you're ever going to be able to have a drug that actually prevents that, because there's nothing to compare it to. So natural history coming out of the registry is huge. The biobank is similar. Where do you think scientists get the tissues and the blood to do the experiments that I just talked about? They need to get it from patients. And in rare patients, you've got to get it from a rare community. You've got to get it from the people that are involved. And education is huge. And I think this goes two ways. One is, I'm here. I know I offer to you that I can educate you about pharmaceutical R&D. This is a 50-minute presentation. I could spend weeks with you if you'd let me, and tell you more and more about it. But it goes the other way too. Researchers need to know what life is like with this disease from the early stages to the later stages. They need to start to understand your point of view as well. So educate them. There's a couple ways you can do that. Write blog posts, put them on CTF. Get involved in conferences where they're speaking, or where they're listening. Tell your story often. In phases 1 to 3, there will be, or you guys probably have already done this. You guys have a lot of the boxes ticked in CTF here, but benefit risk and patient preference studies, they're critical. This is where you tell people what's important to you. Clinical trial protocol review. I saw that there were some opportunities to do that. I did a project with the Friedreich's Ataxia Research Alliance where we taught their ambassadors how to critically review a clinical trial protocol. We taught them what was in it, how it was formed, what were the ethical and the regulatory boundaries that were there, and then we gave them a protocol and said, so tell us about it. And they reviewed it live. That way, CTF doesn't have to rely on staff or other people to do it. They can find volunteers among you. And give feedback about your clinical trial experience. And I'm not thinking about the data that you created that came from you, your adverse events or your benefits that you saw. No, what was your experience? Were you respected? Were you listened to? Was this inconvenient? Was it a burden? What would make it less of a burden? My whole encouragement here is find a way to give that feedback. They may not send you a survey, so you might need to write some notes, or you might make a phone call. We already talked about the one I was going to talk about here, which is patient-focused drug development. Oh, no, I didn't. I haven't done anything around post-approval, but there'll be more studies that'll go on after a drug is approved, and there'll be other drugs that will come along. And you should participate in that. And once again, give feedback. The other big piece that I don't have time to talk about is access. And you're going to have to come together to lean into why should the payers pay for the treatments that you get. So my call to action here is keep learning. Just keep asking questions. I had a boss once who said, never stop asking questions, because I'm really annoying about that. I'll be asking questions during this whole conference. That's my shtick. Keep asking questions, keep learning. Raise your hand. People were asking for volunteers this morning. Great, I'll do it. Can I help somehow? Just raise your hand. Tell your story. And I've been working with the Barth Syndrome Foundation to help them tell their stories in a more effective way, so that they're talking to the audience that they're trying to influence, and they're actually asking for something that can be done about. Build relationships. There's going to be people from biopharma here at this conference. You know what? Sit down, have a beer with them. You don't have to talk NF, you don't have to talk about science. Just get to know these people, because when you want to give feedback someday, maybe that's the person you can say, hey, could you get to the right person in your company? You just need to know people. So just build relationships. That's what the patient engagement folks that are here are trying to do, is how do we build relationships with this large community? Make your voice heard somehow. I mean, it sounds kind of redundant there. And then just participate in trials, participate in I like it, what was it, a drug utilization committee? You need to call that DUC, the NF DUC. And then, just to close my time here, reach out to me. There's my email. Reach out to me here this week. I really do want to get to hear your stories, and check out my website, and we've got some other things that may help you. Thank you very much. This was an honor.
Thank you so much, Kevin. We are getting a little tight on time, so I'm going to ask you the question that was in the app, and because somebody put that in there. It says, if 97% of compounds don't become medicine, some of the audience may be wondering what % of clinical trials in phase 2 or 3 are successful. That's a tough one.
It's actually not.
Oh, great.
Yeah, so there's a study that's done, and it comes out every 10 years or so. The next time it'll come out will be 2024, I believe. It's done by Tufts, and the CISGRIP is the name of the group that's involved in it. And they look at all that. And unfortunately, well, let me do the good part. 80% of what goes into phase 3 succeeds across the industry. It's a little different for biologicals. It's a little different for other types of therapies, but for small molecules, 80% of phase 3 goes through. Unfortunately, in phase 2, you know, when you're trying to prove your hypothesis, about 20% make it. 20 to 40% is the range. And that's where you take the big hit. The problem is when you go from discovery, preclinical all the way through, you have to multiply those failures. So it's like, okay, well, 60% times 50%, that's down to 30%, times 20%, that's down to 5%. You know, you just, it all multiplies on itself. But unfortunately, phase 2 is that area. But I'm not surprised by it, because you're testing a hypothesis. You had an idea, like the guy said in the video, a wild what if. It didn't work. You gave it your shot. Alzheimer's drugs are actually questioning the very basis of what we think Alzheimer's is caused by now, because so many drugs have failed in phase 2 to show an effect using the same theory.
Thank you again, Kevin. We really appreciate this. Yes, thank you. Round of applause.